Hello students, I am Arpita. Welcome to my channel Grow with Biology. Today we'll deal with the topic of labeling of chromosomes. So what is labeling of chromosome? Chromosome labeling means a systematic identification and naming of chromosomes as well as to its regions, to its arms as well as to its band. And this system of nomenclature is known as chromosome labeling. And whenever we do this, we can compare it with the another arms like the normal structures of the chromosomes, and when we compare with those, we can identify if there is any structural abnormalities, if there is any change in the gene location. And this is given by ISCN, that is International System of Human Cytogenomic Nomenclature. This is regarded as the universal standard recorder for the genome mapping. Now, what is its importance? It gives a standardized chromosome maps. It enables global understanding of genetic diagnosis because the normal human uh, chromosome banding patterns has been given. And now when we compare it with the person's or the patient's one or the samples one, then we can identify is whether there is any genetic disorders or not. There it gives in a critical mapping. It helps in analyzing of cancer cytogenesis as well as in karyotyping. Now, what is the basic chromosome structures? If we look at the structure, you will find that there is centromere, which divide the entire chromosome into two parts, the upper part and the lower part. Upper part can be a short arm and be a long arm, or both can be of the same size. At the end of the centromeres, there are presence of telomere. Now, what is the function of this telomere? Telomeres prevent the attachment of chromosomes head to head and tail to tail. So, another chromosome will not get attached to this butterfly-like structure. Single strand or the single part of the chromosome is known as chromatid. Now, if we look at the structure, there is one constriction known as centromere, that is known as the primary construction. The upper arm is known as, or the short arm is known as the P arm, and the long arm is known as Q arm. Now, this P and Q must be written in small letters. Telomeres are at present at the end. Now, the bands which we obtained by karyotyping are light band and dark band. They are light and dark orientations or alternate present in the entire chromosome. There are subdivisions of the bands also present in the chromosome or as observed. Now, banding and numbering system, how is this ISCN numbers guidelines are? Now, first of all, you have to write the chromosome number. Whatever chromosome you are studying, you have to first mention the chromosome number. Then you have to mention the arm. There are two arms, P and Q. So, which arm you are following, you have to mention that arm. Then the region which you are following. After the region, there is position of band and the subband. So everything has to be written in this sequence. Subband has to be written with a decimal or a full stop before it. So number, arm, region, band, and a dot. Then we have to add the subband. Now the numbering convention, the band number starts from the centromere and protrudes outwards. So the band which is closest to the centromere will be numbered as 1, and then as you move towards the telomere, the band number will get increased. Similarly, the subbands also. Subbands are always denoted by the decimals. Now the detailed example we can see that now it is 13q14 means it is chromosome number 13. Arm is Q, 1 is the region, 4 is the band number, and 0.1 is the subband. So this is the location of a particular gene in chromosome number 13. Labeling of a karyotype. So now what is the labeling? The standard human karyotypes are generally arranged in 1 to 22 autosomes, and then the sex chromosomes. They are oriented such that the P arm is always at top and Q arm is at bottom, and the bands are labeled outward from the centromere. So this is the basics of labeling of in the observed. Now suppose this, it is 1q336.3, means it is chromosome number 1. P means the short arm, 3 means the region 3, 6 means the band 6, and point 3 means the subband 3. If we look at 5q21.2, it means chromosome number 5, arm is Q, region is 2, band is 1, and subband is 2. Now we move to the labeling of the chromosome abnormalities, that is ISCN notations. If there is any duplication or there is any deletion, then how it is written. Now it is the normal is 46 chromosomes XY and deletion at 5th. Then the location is Q113 to Q33. So this is the deletion which we have been observed. If there is duplication, then duplication in 7th means 7th chromosomes Q, then 1 that is chromosome band 1 or band 11.23 to Q, that is again there is duplication at 22. Similarly, there is inversion, there is translocation, and there is grain loss. You can see that there is chromosome number 15, there is a normal chromosome 15 and abnormal chromosome. We can see that there is change in the banding pattern in the highlighted regions. Now, the practical labeling in research and diagnosis. In gene mapping, it exclusively helps in gene mapping. Then in cancer cytogenetics, in Philadelphia chromosomes, you can easily find the breakage and translocations, which is a marker for Philadelphia diseases. Then there is prenatal diagnosis in late pregnancies for checking any of the syndromes present in the child. This type of techniques are followed. If we summarize it, we can summarize that suppose the labeling is 1q21.1, that is chromosome 1, long arm Q, region is 2, band is 1, subband is 1. T means translocation of between chromosome 11 to 22, and the region is Q23 to Q11. Deletion 15q means it is a deletion and bands of 11 and 13 on chromosome number 15. Tools for chromosome labeling. What we can use is Carrier Viewer or KIDAS, that is a chromosome visualization tools. UCS Genomes Browser, that is for gene location. Online ICN resources, which is updated every 4 to 5 years. These we can use. So you can plan of your own chromosomes changes. You can write your own think or thought chromosome variations, and then do the practice of writing where what bands, what subbands. You make your own questions and try to solve it out. So finally we can conclude that chromosome labeling is very essential for studying of cytogenetics, so that we can get the morphology, we can observe the banding pattern as well as the standard nomenclatures. We can compare it so that we can identify whether there is any diagnosis of diseases or not. It is used in research, it is used in diagnosis of diseases, it's used in clinical genetics. So the mastery in ICN follows always allows professional to accurately describe the chromosomal loci and abnormalities with precision. So this is a very specific technique, very updated techniques, and very knowledgeable techniques. If you can get a chromosomes, you can study a chromosome in these levels, you can identify or get a knowledge of many lots of things which we can't study from a normal chromosomal studies. So thank you for being with us. Hope this will be helpful. If you have any queries, you can write in the comment sections. If you want any other topics to be understood, you can definitely mention in the comment section. Thank you and happy study.